Greetings, and welcome to Today in Science. It has become clear in recent years that one of the biggest hurdles modern science faces is the understanding and treatment of Alzheimer's disease. According to the Alzheimer's Association, Alzheimer's is the sixth leading cause of death in the United States, and it can be expected to cost the country up to $1.1 trillion annually by the year 2050 as the population continues to age. Deaths from Alzheimer's disease are up by 89% since the year 2000, and it is clear that this number is only going to increase. So, what actually is Alzheimer's disease? Alzheimer's is a form of dementia, or decline in memory and cognitive function, that accounts for an estimated 60 to 80% of all cases of dementia. Alzheimer's is a neurodegenerative disease, meaning it worsens over time, and there is no cure. The exact cause is unknown and the subject of much current research, but it is believed to be genetic in origin and related to the buildup of plaque in the brain from a protein called amyloid beta. The formation of this amyloid plaque results in a loss of structure and function in neurons, leading to the characteristic cognitive decline that defines Alzheimer's disease. The amyloid beta protein comes from another precursor protein, conveniently called amyloid precursor protein, or APP for short. The actual location of the APP gene is found on chromosome 21, the same chromosome that is abnormal in trisomy 21, better known as Down syndrome. This explains why nearly all people with Down syndrome exhibit at least some symptoms of Alzheimer's disease by the age of 40. An extra copy of chromosome 21 leads to more APP and therefore more amyloid beta, which means more dangerous plaque. Treatments do exist to combat the disease, but they are often more expensive and less effective than many doctors and patients would like. Just earlier this year, pharmaceutical giant Merck ended clinical trials on what seemed to be one of the most promising Alzheimer's treatments, Verubecestat, after the data showed virtually no chance of working properly. The British Medical Journal described Verubecestat as a promising candidate in a field littered with failures. Maybe not so much. It goes without saying a little bit of progress in the realm of Alzheimer's research is much needed. Luckily, one new study shows what might be great potential for the future of treatment of the disease. A study published in the Annals of Neurology on June 9th of this year has found an association between the neuroimaging biomarkers of Alzheimer's disease and a loss of one's sense of smell. So, what exactly are neuroimaging biomarkers, and what are they for Alzheimer's disease? Biomarkers are the way that scientists and doctors are able to diagnose someone with a disease. Because it isn't feasible or even remotely safe to actually open up the skull of each and every patient suspected of having Alzheimer's disease and examine their brain, specialists will perform various scans and tests to provide them with enough information to make an accurate prediction about what exactly the pathology is and how they can best treat it. These tests consist of scans like MRIs or PET scans. Magnetic resonance imaging, shortened as MRI, is a type of brain imaging most useful for mapping structures in the brain. An MRI machine works by passing a huge amount of electricity through coils to create a magnetic field, which then causes the water molecules within our body to act as magnets that can be measured by a scanner and then interpreted by a radiologist to create a somewhat detailed image of the brain. Positron emission tomography, shortened as PET or PET, is another type of brain imaging that is best for measuring energy usage and activity within the brain, rather than the brain's structure, like an MRI. Glucose, the sugar that the body uses to provide energy and carry out everyday function, is taken up by the various organs and cells of the body so that they can continue to work properly. Measuring glucose uptake in a certain region of the body provides insight into how exactly that area is functioning metabolically and whether or not there is some sort of dysfunction in the area that causes an irregular glucose uptake. Because there is no way to magically sense glucose uptake within the body, a radioactive form of glucose that emits measurable gamma rays, called fluodeoxyglucose, is given to a patient and absorbed into their body and bloodstream. Doctors can then monitor the pathway FDG takes throughout the body and diagnose any malfunction. MRI scans of Alzheimer's patients show abnormal thickness of the cortex, the outermost layer of the brain, and a smaller hippocampus volume, a key structure in the brain responsible for long-term memory. PET scans of Alzheimer's patients show a lower amount of metabolic activity in the brain than would be expected, implying decreased neurological function. Scientists have previously linked certain neuroimaging biomarkers with the onset of mild cognitive impairment, an early symptom of Alzheimer's disease. Scientists have also previously linked olfactory impairment, or a decrease in the ability to distinguish smells, to cognitive decline. Researchers in this study sought to establish whether or not there was a link between the neuroimaging biomarkers of Alzheimer's disease and olfactory impairment. They carried this out by administering a brief smell identification test, or BSIT, to 829 participants with ages ranging from 70 to 89 years old. The same participants had an MRI performed to test for abnormal cortical thickness and reduced hippocampal volume. A total of 611 participants had PET scans performed. The BSIT test consists of 12 scratch and sniff smells such as pineapple, lemon, smoke, and soap. The subject is asked to scratch and sniff a smell and then is given 4 possible options to pick from of what the smell actually is. One's BSIT score is merely the sum of all their correct answers, ranging from 0 to 12. What they found was that there was an association between BSIT score, olfactory impairment, and neuroimaging biomarkers for Alzheimer's. A linear regression was calculated, comparing olfactory impairment to MRI results, and found a significant relationship between the two. The scientists who performed the study were 95% confident that the slope of the linear regression line was between negative 0.09 and negative 0.76, with a p-value of less than 0.01. What this basically means is there's less than a 1% chance that these results occurred by random chance. MRI and PET scans are expensive, somewhat invasive, and inaccessible to many people. Alzheimer's often goes undiagnosed for far too long because of these barriers, and because of its neurodegenerative nature, this damage is irreversible. The best line of defense we have against Alzheimer's right now is prevention, and creating a simple, easy, non-invasive, and inexpensive way to test for the disease could mean the difference between proper treatment and rapid cognitive decline for many patients. Linking issues with smell to elaborate, expensive neuroimaging means a simple BSIT can easily replace a PET scan and open up the door to early diagnosis in many individuals. It should be noted that this is only one study, and it doesn't definitively prove anything about the relationship between smell and one of the most deadly diseases on the planet, but it does shed some light on a potential for future treatment. The study reference can be found linked in the description, along with a few other links to learn more information about Alzheimer's disease. Thank you for watching, and I hope you enjoyed.